It's time for another amazing chemistry video with Mr. Stapleton. Proudly sponsored by Farming Doing Nice Coffee.
Hi guys, welcome to the next video. This one's going to be all about proteins. Now a couple of things before I get into it. Number one, I'm actually trying out some different pens today. So I'd really like to get your info, your feedback around how you find these ones, right? If you think they're better or worse than the previous pens I was using. Obviously they're newer, so they're going to probably show up a bit more brighter. Just let me know how you like them. Secondly is make sure before you jump into this video that you've watched the one on amino acids, just so you understand the general structure of an amino acid, okay? And that you can understand what happens with the carboxylic acid and the amine functional group, because that will come into play importantly when we're looking at proteins today. So what I'm going to be talking about is the four main structure, or I'm really only going to go into depth on the first three main structure variations within a protein. So remember, I'm only going to be looking at this from a chemistry viewpoint, and so in chemistry we're obviously concerned mostly about bonding and secondary interactions between them. Okay, so I'm going to go through, and the primary structure of a protein is what I'm going to start off with. Okay, now what you need to understand is that protein is basically the building block of all the things we need inside our body, and it happens by having amino acids linking to each other. Okay, and this is what the primary structure is all about. So if you remember from the previous video when I was talking about an amino acid, okay, so I've got a carbon, a hydrogen, we've got our R group here, and we've got NH2 and our carboxylic acid functional group here. What I'm going to do is I'm going to react that with another amino acid, okay, so I'm going to add that to another amino acid over here. I haven't left myself enough room, sorry, so I'm just going to try and squeeze this in if I can. There we go. Alright, and what I've deliberately done is I've put the carboxylic acid functional group at one end next to the amine group of the other one. Now, two amino acids can link together via condensation polymerization. Now, condensation polymerization is where two molecules join together and they eliminate a small molecule. Now, normally it is water, which is what the case is here. It doesn't have to be though. So what happens is that the hydrogen here from this amino acid actually comes off. I should use a proper marker on this. So that hydrogen actually comes off of here, all right? And so does the oxygen. So that's the OH gone, and we actually lose a hydrogen from over here. So that comes out as a water molecule, H2O. And that came from the OH that was here and one of the hydrogens there, okay? Now what we get, and this is the same as if we're forming an ester, so I'm going to go through this reasonably quickly. So what we get is the first part of our protein now, over here. There's a carbon double bond oxygen, over here. And then that bonds to the other amino acid here. Alright. And you can see what we've done here is this is the start of a protein. Now a protein can have hundreds and hundreds and hundreds of amino acids all linked together, okay, and the order that they go in determines a lot of the important codes for within our body. That's all in biology, I'm not going to go into that too much, but what we're concerning ourselves with here is the firstly condensation reaction that's occurred, where we've taken an amino acid and another amino acid, we've eliminated a water molecule and we've formed part of our protein over here, and the groups that we've actually made, okay. So this group here is called a peptide group. Okay, that's the term when we're talking about biochemistry. If we're looking at it from a chemistry point of view, we also call it an amide group. Okay, and then this bond within here is called a peptide link. So sometimes you'll hear proteins referred to as polypeptides. And the reason for that, the word poly means many, so it's many peptide links or many peptide groups, which is what you're going to get, because if you have a look at this amino acid we've got here, okay, you can see we've got an NH2 group on this end, we've got a carboxylic acid group on this end. So what we could do is bring another amino acid here and react it on with a condensation reaction. We can bring another amino acid here and react it that way. So we can build a polymer chain or a protein chain out all the way like this. And that's the process of the primary structure, is how our amino acids all link to each other to form a long protein chain. So that's the first part. Second one we're going to get to is where we start getting multiple chains together and how they interact with each other. And that's called the secondary bonding. Okay, so what I'm going to talk about next is the secondary structure of proteins. So the primary structure that we just looked at was how the amino acids actually link to each other. Now what you get, and I'm going to try condense down the structures a little bit to show you, right, is you're going to get two protein chains and then they start to link next to each other. So here's our first protein chain. So we've got our R group there, hydrogen, C-O off that way, nitrogen, hydrogen. There's our peptide group, right, or our amide group, over to another carbon. There's a different R group, hydrogen, nitrogen, hydrogen, another peptide group. Okay, and you can see we're just building up the chain of our amino acid. All right, and I'm just going to end it off at that. So that's just one section of an amino acid. What we do is, if there's another section of the amino acid, okay, next to itself, all right, we can actually get some bonding. So I'm going to draw another one here, and I'm going to kind of flip it or invert it a little, or shift it a little bit, I should say, so that you can actually see the interactions that we get. So I'm going to start actually right here, and hopefully you'll be able to work out straight away why I've gone there. So that should be nitrogen, hydrogen, carbon, hydrogen, another R group like that, another carbon, another R2-5. Alright, so we've got these two distinct chains here, and what we're really concerned about when we're looking at the secondary structure of the protein is how it bonds to itself. Okay, so if you're doing biology, you might hear things talked about called alpha pleated sheets or a beta helix of a protein. DNA is a good example where you get that spiral, that helix structure. But that just is something that occurs when you get bonding between parts of the chain. And so what can happen is that you actually get some hydrogen bonding occurring between your two protein chains. Okay, so what I'm going to show you is here we have a delta positive hydrogen, a delta negative oxygen. So we actually get a hydrogen bond occurring between them. We've got another one here and another negative there, so we get some hydrogen bonding occurring there. But obviously we've got delta positive there, so we get hydrogen bonding down here. We've got a delta positive there, so hydrogen bonding this way. One here, hydrogen bonding down here. These oxygens up here will hydrogen bond as well. So what you can start to get occurring is all of this hydrogen bonding occurring between the chains, okay? And that hydrogen bonding between the chains helps to get it to linking together. So it can either fold over on itself, okay, if that's one way the chains line up, or they can actually wrap around themselves as they bond as well. So that's the two types of structure types you can get. And it's purely due to hydrogen bonding between the amide groups of the protein chain. Now, that is different from the tertiary structure of a protein, and we're going to really get into the tertiary structure of a protein because that's really going to help us to understand why enzymes, which are proteins, get denatured under pH or temperature conditions. So I'm actually going to stop this video here, and the next video I'm going to do is going to be all about tertiary structure of proteins, just because I don't want it to go too long, and explaining about denaturing of enzymes as well. So if you've got any questions about this one, ask. But otherwise, maybe have a quick look, make sure you understand primary, which is your bonding of the amino acids in the sequence to form your protein, and secondary, which is where you've got your chains that fold over or wrap around themselves due to hydrogen bonding between the very polar carbonyl and amine groups of your protein chain with hydrogen bonding, which are quite strong, so they hold the protein in nice and tight. All right. As always, ask any questions, otherwise maybe go on to the next video when you're ready. Thanks guys, see you.